Historically, we have used natural sources of water to meet our needs. But as the population of the Earth increases, we are going to start to rely on new materials and alternative ways of making clean water to quench our growing thirst.
Hi, my name is Gabriel Montano, and I'm a professor of applied physics and materials science at Northern Arizona University. I'm going to introduce you to biomimetic membranes and current and future technologies. What are biomimetic membranes? Why do we care about them? And what can they do? Nature uses membranes to compartmentalize, create barriers, perform complex functions essential to life. Not only are the exteriors of cells membrane architectures, but all the internal compartmentalization that creates cellular function consists of membranes. These membranes are based upon amphiphilic molecules called lipids. A lipid is an amphiphilic molecule, meaning that it has both water-loving and water-hating portions. When placed in water with other lipids, they naturally form membrane structures, with their water-loving head groups pointed out of the cell and typically into the cell interior, where the water is present as a solvent. The water disliking tail groups point inwards towards each other and create an oily interface that excludes water. The result is a barrier, one that separates the external and internal environments. This simple architecture is responsible for countless biological functions, without which life would not be possible. As an example, the membrane in plants is where photosynthesis happens. Photosynthesis is actually a biological machine. Light is absorbed, water added, and oxygen and fuel for the plants are formed. Photosynthesis typically occurs in specialized membrane structures called chloroplasts. The multiple membranes perform two primary functions. First, they hold the protein complexes responsible for photosynthesis in close proximity to one another. And secondly, they create the potential for a gradient where charge can be built up on one side versus the other of the membrane. Like a battery, this charge buildup drives the formation of energy and food. The process results in oxygen and hence life. So what is a biomimetic membrane? Traditionally, biomimetic membranes are composed of lipids. They can incorporate proteins and other biological or synthetic nanomaterials in efforts to typically replicate nature or improve upon it for some functional output. In the 1960s, liposomes were discovered. Liposomes are artificial cell membranes consisting typically of lipids that mimic the cell membrane architecture found in nature. The discovery of liposomes launched the field of biomimetic membranes, which has furthered our understanding of important cellular processes such as cell communication and complex processes such as photosynthesis. In addition, new technology platforms for sensing, optics, communication, and even quantum computing are being sought using membrane technology. Liposomes allow us to study complex processes such as cell communication and drug targeting. As an example, here we see an artificial photosynthetic power plant in which a liposome is used to house synthetic organic chromophores that when hit with light cause protons to move from one side of the membrane to the other via a process called charge separation. This is the same way that plants utilize light. The result is a proton gradient where more protons sit on one side of the membrane relative to the other. In this system, a biological protein powerhouse, ATP synthase, is also incorporated. The proton gradient enables the ATP synthase, which is like a windmill, to function producing ATP. This simple construct mimics the natural process of photosynthesis. The membrane here is vital for two reasons. First, it creates a matrix to organize the cofactors, the organic molecules and the ATP synthase. And secondly, it enables the generation of a proton gradient, necessary to drive the ATP production. The result is a simple yet elegant replication of one of the most important processes ever developed that is responsible for life as we know it, built upon a biomimetic membrane system. Supported monolipid bilayers are another biomimetic membrane architecture, in which the lipid bilayer is supported on a surface. Such systems create an entirely new membrane architecture that can be exploited not only to study biological processes, such as membrane protein function, but used in fields ranging from photovoltaics to energy production and drug discovery. Here we see an example of a biomimetic membrane that is on a supported glass substrate. Dyes are incorporated into the membrane so that we can visualize it, and that results in the red and green colors that you see. A pattern is created here using a natural biological molecule called lipopolysaccharide in a chemical treatment. In nature, this molecule is responsible for septic shock, but for material scientists like myself, it's a means to create chemically modified, defect-free micro-nanopattern membrane arrays of exquisite symmetry and limitless dimensions. When we add this to our membrane systems and control surface factors, we can create ordered photonic arrays. The result is a supported membrane system with unique photonic properties on a scale smaller than the width of a human hair. This is an example of a membrane-based photonic material for applications in optoelectronics, sensing, and quantum computing. In this video, artificial membranes are used to investigate an important cell communication process involved in stem cell differentiation. In this example, supported membranes are used to better understand this important biological signaling process. This is a pattern membrane array that hosts different proteins in it, and when a cell interacts with it, it triggers different cellular functions. That's what we're observing by the colors that you see here. This is an example of biomimetic membranes advancing medical science, an important application process of biomedical membranes. This research even expands beyond simple membranes to tissue engineering. So what are the tools that are used to study biomimetic membranes? Biomimetic membrane materials investigations are inherently interdisciplinary and use tools from a lot of different traditional sciences. Advances over the last few decades, particularly in regards to surface techniques, have quickly advanced findings. As an example, scanning probe microscopy, and in particular atomic force microscopy, allow for molecular level characterization. Here we can see different membrane proteins with high resolution structure using atomic force microscopy. In these images, each circular component is an individual protein, and thus we're actually visualizing individual proteins with extremely high resolution. And you can not only see the structure, but oftentimes the dynamics of these intricate biomachines. The supported lipid membrane enables new techniques such as AFM that result in these type of high resolution techniques.
So I think that people underestimate the complexity and behavior of cell interfaces and membranes. Oftentimes we think of cells as their own individual lipid membrane and bilayer structure, but there's a lot of complexity that goes into the interaction and behavior of cells with not only each other, but also their surrounding environment, be that proteins and tissues or any sort of material or surface that they're deposited on.
More recently, other molecules such as polymers have been used to mimic biological membrane structures. These membrane materials look and act like lipid-based membranes, but with added advantages. They are more robust, more dynamic, and have a higher potential for device integration. Here is an example of pattern membrane arrays created using amphiphilic block copolymers. These polymers have water-loving and water-hating groups just like lipids that allow them to form membrane structures just like lipids, but they're much more robust and easier to integrate in different types of devices. In addition, we can use these to create artificial organelles. They've been created using these amphiphilic block copolymers, and in this case, mimic the structure and function of biological systems, ushering in a whole new era of biomimetic membrane materials. These membrane materials look like cell membranes, they act like cell membranes, but they're completely synthetic in nature and have a lot of advantages to biological systems. Here we see a chloresome, which is a photosynthetic light harvesting complex, responsible for absorbing light to create energy. It's a large membrane structure where the lipids form an outer coat that allow for tens of thousands of light-absorbing molecules to organize to absorb light for photosynthesis. This is one of the most efficient photosystems ever known. Some of the bacteria that utilize these systems may see one photon every 6 hours, so every photon must count. if they're to be active and survive. Using polymers, we have created a completely synthetic chlorosome called the polymer chlorosome nanocomposite, or PCN, that's shown here. It's not only as efficient as that of the natural component, it has new properties, what we call emergent properties. Instead of just a single color, we can tune these to an array of colors. In addition, we can infer new properties on these and make multifunctional types of chlorosomes. In this case, what you're seeing is a magnetic PCN complex. We've actually incorporated magnetic nanoparticles, which you can see here in the rust color looking objects that sit at one end of the chlorosome. With these, we can manipulate the chlorosomes, we can, by applying magnetic fields, and create new types of functional responses out of these systems. Biomimetic membrane research not only leads to scientific discovery, it is inspiring real-world applications. And the understanding and implications of membrane technology are being used to save lives today. As an example, the N95 mask is based upon the active transport mechanisms of membrane technology, critical to preventing the spread of COVID.
We're here with Dr. Peter Tsai, who's the inventor of the N95 mask, and asking him a few questions about the mask and also his inspiration. Can you tell us a bit about what your inspiration was to the N95 mask, and what led to exploring this idea of electrostatic filtration?
So I work at the University of Tennessee from 1980s, and we had microfiber process to study the process and to study the application, and also to study the raw material for the process. And because this is microfibers, so we know that microfibers can be a good material for air filters. And we also figure out if we can put the charges into the fibers, that we increase the transmission efficiency. Because it is made of a fibrous material. So there are pores between fibers. So that is easier for the air to pass through. And the efficiency comes from the surface of the fiber to attract the particles. And if there are charges in the fibers, in addition to the mechanical attraction, there are electrostatic attraction. In this case, for this kind of particle size, that is sub-micron, and more precisely, it is 0.075 micron, the particle size for the N95 testing. And at this kind of inhaling or exhaling air volume, this kind of particle size and this kind of velocity, the electrostatic attraction is 10 times higher than the mechanical attraction. So in 1992, I invented the electric charging technology, and this charging technology is called corona charging. Okay, the same corona as the coronavirus. This is another coincidence. Okay, but this corona charging has nothing to do with the coronavirus. But the corona means a halo or something around something. So, you know, see, coronavirus is something like a crown on the surface. You know, see, when we see the SEM image, we see the coronavirus looks a beautiful crown. So that's called coronavirus. And my corona charging, because at dark, you will see a very dim light around a wire or a needle that generate corona discharge. Okay, so we call it the corona discharge. Then this filter was originally to be used for air filters. Then after that, it's because this my invention in 1992, but in 1995, US is going to issue another new specifications for the respirators. So industries work with me to see if my technology can meet the specifications of the new standard.
So we have seen how manipulating the charge on the fibers of the N95 mask using a corona discharge can help create a membrane that not only captures the coronavirus, but does so while allowing the user to still breathe easily. We have also seen how nature designs biological membranes and how we can mimic what we learn in our own membrane design. Throughout human history, we have been using these principles even when we did not comprehend the underlying science. Now that we have a deeper mechanistic understanding of why these membranes work, we have solved many of today's challenges that involve membranes. How will membranes that use nature for inspiration be used to help address future challenges?